Hi everyone and good morning. Today we are going to cover topic 6.2 leaf structure. Today we are mainly going to discuss about the features of the leaf that is adapted for photosynthesis. And your first learning objective today is 6.2.1. State that most leaves have a large surface area and are thin, and explain how these features are adapted for photosynthesis. So the first step to success is to state how leaves are adapted for photosynthesis, and then state the advantages for those adaptations. But before we talk about these adaptations, let's just do a background on the plant itself. So all plants have a root and shoot system. The shoot system and root system can be seen in this diagram right here. And the shoot system is basically the parts of the plant that is above the ground, and the root system are parts of the plants that are underground. The shoot system has a main function, which is to carry out photosynthesis, and the root system's main function is to provide water and nutrients for the shoot system. And so the root system has a main function of absorbing some minerals and water. And this is actually to help the leaves carry out photosynthesis. They're going to absorb water and also minerals like nitrate ions and magnesium ions. The plant shoot system consists of the stem and the leaves. And as you can see here, the stem actually is the one that helps to also carry these minerals like magnesium ions and proteins and even water to the leaves. The shoot system has the main function of basically carrying out photosynthesis. The leaves will take in carbon dioxide and also sunlight. Carbon dioxide is a raw material of photosynthesis, and sunlight is where the plants get the energy to actually carry out photosynthesis. So the majority of plants and leaves carry out these jobs. Firstly, so just the leaves, they take in carbon dioxide, they take in sunlight, they manufacture carbohydrate, that means that they make carbohydrate, and they also release the waste product oxygen. So there are these four main functions of the leaves. So how do they take in carbon dioxide is that they have these structures, the guard cells, that actually can be found at the bottom of the leaves. And guard cells, what they do is that they can open what we call the stomata, which is basically a hole underneath the leaves so that the carbon dioxide can actually enter the inside of the leaf. The leaves also can take in sunlight, and how they do this is that they have these organelles, the leaf cells. Some of them have these organelles called chloroplasts, which contain a green pigment called chlorophyll, and they have the ability to absorb sun energy. They manufacture carbohydrate, and what we mean by this is basically the definition of photosynthesis, which is making food, and food is in the form of glucose, and that is what we mean by manufacture or making carbohydrate. And this carbohydrate, glucose, can actually also be converted into cellulose. And if you recall, cellulose can be mainly found in the cell wall because cell walls are made up of cellulose. And cellulose is also a fiber. Leaves can also convert the glucose into starch for storage. The waste product oxygen is basically what you get after photosynthesis occurs. And the leaves also try to remove this waste product. And the feature of the leaf that we're looking at here is also the guard cell. The guard cell has a stomata, which is a hole, and the oxygen, the waste product, can actually get out of the leaf and be removed this way. So if you see over here, the guard cells are involved in mainly gas exchange. They exchange carbon dioxide coming in, and they also remove the oxygen. They want to get it out of the leaf. So here is the external features of a dicot leaf. Dicot stands for Dicotyledonous. and we'll talk about that some other time about dicots and monocots, but today I want you to basically just learn about the features of a typical leaf. So when we talk about features, we're always talking about how it looks, what does it have. So here we have right near the leaf, this is the leaf right here, and right at the edge of the leaf you can find a bud that is a mini baby leaf that is about to grow, and you call that a bud. And the thing right here that actually holds the leaf upright, like literally holding it up, is called the petiole. Then right here you can see the edges of the leaf, yeah? This edges is what we call a margin. So if you imagine if it is a circle, so you learn the shape of the circle in maths, and the circumference of the circle, that is what a margin is basically the outline of a leaf. That is the margin. And here in the middle, the petiole is this part, but in the middle of the leaf, we call that the midrib. And the midrib branches out into branch veins. And also they have these little vein networks. There are even more smaller branches. And the whole leaf right here, the surface area of the leaf is what we call the lamina. So for example, if you're looking at a circle, here we're talking about the area of the circle, which is pi r square. And the circumference of a circle, that is what we mean by the margin. But instead of a circle, we're talking about the leaf here. So the lamina is literally this whole area, the area of the leaf. So adaptations of leaves for photosynthesis. Although there are many types of leaf shapes, as you can see here, a lot of the shape of the leaves are pretty actually different. However, most leaves have these features. First one is a large surface area, which means they have a broad lamina. Remember, lamina is the area of leaf. And second feature is that all leaves, or most leaves, most of them are very thin. So right here I have a video for you guys, and this is what we mean by large surface area and thin. So this is the how thin leaves are. They are very, very thin. And if you have a look here, that is the surface area. It is pretty broad. It's not usually small, but it is pretty broad. So this right here is the lamina of the leaf, or the surface area, and it's pretty large. And also the thickness of the leaf, it is very, very thin. So these are the two main features, basic features of leaves. But how does that really help them in photosynthesis? So having a large surface area, the lamina of the leaf is broad and flat in shape, and this gives that large surface area, and this helps the leaf to absorb a lot of sunlight. That means that when the leaf is broad, that means that they can receive, and a lot of area can reach, sunlight can reach a lot of the area of the leaf. Another thing is also that when the lamina is large, then that means that we can have a lot of these guard cells at the bottom of the leaf. And if they cover a large surface at the bottom of the leaf, that means that they can absorb more carbon dioxide. Leaves are mainly thin so that carbon dioxide can diffuse across a short distance to reach the inner cells. So literally that just means that there's a distance for diffusion, very, very short. And that means that the leaf cells can actually receive carbon dioxide faster. So let's look at the cross-section of the leaf. When we're talking about the cross-section here, we're talking about cutting the leaf like this. Imagine like you're cutting like tapak kuda. That is what we mean by cross-section. So here we have cut a cross-section of a leaf, and you can see how actually quite thin it is. And if we look at under the microscope, this is just a drawing. Actually, the leaf itself have many layers of cells. So they have different types of tissues that have their own specific function. And now we're going to actually talk about those layers and the different tissues that you can find in the leaf, and about how they are adapted to photosynthesis. So this is your new learning objective. 6.2.2 Identify and label the cuticle, cellular, and tissue structures of a dicotyledonous leaf as seen in diagrams or photomicrographs, and explain how these structures are adaptations for photosynthesis and gas exchange. So our new step to success. Firstly, identify and label structures of the leaf. That means that if you are able to look at that structure and be like, yes, I know what that is called, then that you have achieved your step to success, the first one. Second step to success is to state how these structures are actually involved in photosynthesis and gas exchange. So talk about the adaptations, how they are very helpful, especially in photosynthesis and gas exchange. So let's look at the cross-section of the leaf over here. It's very, very detailed. You do have a few layers. So let's start off with the top layer. That line over there is called a cuticle. And if you look at the bottom here, you can also see that they also have a cuticle at the bottom of the leaf as well. And the first layer of cell here is what we call the upper epidermis. Second layer here, the ones that look pretty similar, they are longish in structure, and this is called the Palisade mesophyll layer. Here we have the spongy mesophyll layer, which also you can find around here. So those circular like cells, so these are plant cells, although they are circular in shape, they're still what we call regular because they look similar to each other. These are called the spongy mesophyll layer. And right here you can see that inside of the leaf they also have air spaces, and they are called intercellular air space. The reason being is because it's in between the cells of the spongy mesophyll, so we call this the intercellular air space. Over here in the middle we have these red color kind of like cell-like structures called xylem vessels. And at the bottom is the phloem, and they both make up the vascular bundle. And right at the bottom here is called the lower epidermis, so this layer here, and you can see your guard cells are part of the lower epidermis as well. And in the middle of the guard cell, when it is open, you can see the hole in there, and that is what we call the stoma. If we're talking about plural, a lot of them, we call that the stomata. So this right here, this is just a drawing, isn't it? But in reality, this is what it looks like. This is a Picto Micrograph. It means a picture that you can get from the microscope. And this is actually what we can find in the school. So I got this picture from the microscope in school, and we can see that this is a comparison. So at the top layer right there, very, very thin, that is the cuticle. And then you can see this one right here, the layer of cells that are kind of like lying down, and that is your upper epidermis over here. And right here, there are these cells that are quite long, and that refers to this Palisade mesophyll layer. And in the middle, this big part is actually here, the vascular bundle. So the red ones over here, that's your xylem, and at the bottom here should be your phloem. And here we have circular cells, and there are air spaces right here and here. So this one in this layer is actually your spongy mesophyll layer. We can also see the lower epidermis, but I think we can't really see the guard cells anywhere here, but there is a faint layer of line right there referring to the cuticle, just right below the lower epidermis. Alright, so let's start with the cuticle first. What is it? It is a waxy, non-cellular covering to the leaf. And the function of the cuticle is that it is waterproof so that water does not escape. We don't want water to escape because water is a raw material for photosynthesis. It is transparent to light, that means that light can enter the leaf and reach the photosynthesizing cells of the leaf. It provides some protection from mechanical forces as well. So in drawings, it is just those two straight lines. So what we mean by being transparent is that, having it transparent in color so that they can allow the sunlight to pass through down here. That light can pass through the photosynthesizing parts of the leaf. Photosynthesizing parts means that the ones or the cells that actually contain the chloroplasts. So you can see here that the Palisade mesophyll and the Spongy mesophyll and also the guard cells contain chloroplast. So they are the photosynthesizing cells of the leaf. The upper epidermis is that layer right there. It is a single layer of cells, and their main function is to secrete the cuticle. They have no chloroplast. That means that they can allow light to enter the cells beneath it. The Palisade mesophyll layer is a layer of one or two, depending on the species. Some can have 2 layers of Palisade mesophyll. They are closely packed and elongated cells with a water film coating their walls. So water film is this blue color right here. And the water film is very important. It's pretty much similar to the water film that we can find in our lungs at the alveoli, and it helps to dissolve gases. So here it helps to dissolve the carbon dioxide that can enter the leaf cell, the leaf cell. Okay, the function of Palisade mesophyll is to receive light, and they have the greatest concentration of chloroplast. That means that they carry out the most photosynthesis. So the Palisade are these ones right here, elongated cells. So just to talk about the chloroplast itself, the chloroplast is an organelle that can be found in leaf cells that carry out photosynthesis. On the outer membrane and the inner membrane, so that means that there are 2 membranes of the chloroplast. This is very, very detailed, by the way. You're going to learn this in 6th form, I think, but you don't need to memorize this for O levels. I'm just here to introduce this to you a bit earlier. So the chloroplast itself has an outer membrane, an inner membrane, and also their own cytoplasm called the stroma on the innermost layer. And in the stroma are these things called thylakoid, or these structures called thylakoid membrane, and this is where chlorophyll can be found. So that means that the thylakoid structures are where light is captured. And when water is transported here, and also carbon dioxide, this is where they can make the glucose during photosynthesis. And this glucose can be converted into cellulose fiber, which is important to make the cell wall of the plant leaf cell. And also, they can also be stored as starch. So chloroplast is an organelle that can be found in the cytoplasm of plant cells, and they contain green pigments called chlorophyll. The main function is to carry out photosynthesis, and it provides a large surface area for the uptake of carbon dioxide too, because you have a lot of chloroplasts. They can only be found in the cells that carry out photosynthesis, and they are the main location of photosynthesis. So chloroplasts can convert glucose into starch and also store it temporarily. That means that if we are doing the test for starch, we can mainly find the starch where cells actually have chlorophyll in them. In dim light, these chloroplasts itself, they can also move. So they are not just stationary, they can actually move nearer to illuminated surface of the cell. That means that parts of the cell, for example, if there's more light here, then the chloroplast will move to the right. They can also convert glucose to sucrose for transport in the phloem. Okay. Now that we learned about the chloroplast, we're going to talk about the other structure, so the tissues of the leaf that actually contain them, which is the spongy mesophyll cells. Spongy mesophyll cells are cells with fewer chloroplasts. They have a few, but remember the most are in the palisade. They are loosely packed. That means that they are not tightly packed like the palisade. They are loosely packed, and the reason for this is that they can create this intercellular air spaces in between them. The walls are coated with a water film. Those parts right there that I've labeled are called the intercellular air spaces. However, over here, I just want to clarify is that in this picture, we have the xylem and the phloem, but over here we don't. So if they don't contain the vascular bundle, then that would be an airspace. So I think I should have labeled it like around here to make it more easier to follow. Okay. So the main function of the spongy mesophyll cell is to carry out photosynthesis, and they also allow gases to diffuse freely through the leaf. So we learned about two mesophyll cells, layers, tissues, basically, mesophyll tissues. We have the palisade and the spongy cells, and they both are actually photosynthesizing cells, and they are found in the middle of the leaf. They are referred to as the mesophyll cells or mesophyll layer, and together their walls provide an enormous surface area for gaseous exchange as they absorb carbon dioxide and release oxygen during photosynthesis. Okay, now let's look at the middle part of the leaf, which is the vascular bundle that is found in the vein of the leaf. So what is the vascular bundle? The vascular bundle is basically the vein of the leaf, and it contains two types of tissues, the xylem and the phloem. At the top right there, that is the xylem, and they are characterized by these very thick walls, and at the bottom there is the phloem. The phloem also contains these living cells called the companion cells. So what are their main function? The xylem strengthens the leaf by helping to resist tearing and transports water and ions to the leaf so that they can carry out photosynthesis. And the phloem would transport the sugar that the leaves make in the form of sucrose, mostly away from the leaf, as well as amino acids, both from and to the leaf as required. And now lastly, we're going to look at the bottom of the leaf, and this is a Picto Micrograph that I also took from the lab, and this shows the bottom of the leaf and actually the lower epidermis. So the lower epidermis is a layer of cells, and they also contain these specialized cells called the guard cells. So you have one there and another, and they come in pairs, and you can see that there is a space in between right there, and that is what we call the stoma. So the lower epidermis, what is it? It is coated with a thin, waxy cuticle. They don't have chloroplasts except for the specialized lower epidermal cells called the guard cells. Guard cells are specialized epidermal cells that occur in pairs, and each pair controls the opening and closing of the stoma, aka stomata, if you have a lot of them. The function of the lower epidermis is to form a lower protective boundary of the cell, and the guard cell's main function is controlling the opening and closing of stoma. And this actually is included in allowing gas exchange to occur. So we have finished both our learning objectives, so let's do a recap of the step to success. Firstly, we had to state how leaves are adapted for photosynthesis and talk about the advantages of those adaptations. So leaves, mostly all leaves, have a large surface area. The lamina of the leaf is broad and flat in shape, and the large surface area is good to help the leaf to absorb sunlight and also carbon dioxide for photosynthesis. Leaves are also thin, so that the carbon dioxide can diffuse across a short distance to reach the inner cells. And now we're looking at this learning objective, and let's do a recap of basically this whole part, which is the structure of the leaf tissues. So at first we looked at the cuticle, and it is this waxy, non-cellular covering at the top and the bottom of the leaf. It is waterproof and transparent so that light can pass through, and it also provides this protection of mechanical forces. Epidermal layer, you have an upper and lower. The upper epidermis is a single layer of cells. They secrete the cuticle, and they don't have any chloroplasts so that they can allow light to enter the cells beneath it. The lower epidermis, it has two types of cells, the epidermal cells and the guard cells. Epidermal cells are pretty similar to the upper ones. They're a single layer of cells. They secrete the cuticle, and they also don't have any chloroplasts. And the guard cells is a specialized lower epidermal cell. They help to open and close the stomatopore so that exchange of gases can occur. And they also have chloroplasts, and they carry out photosynthesis. The mesophyll tissue, you have two types, which is the Palisade and the Spongy. Palisade carries out photosynthesis, and it has the greatest concentration of chloroplasts. It is one layer or two, depending on the species, closely packed, elongated, and they are the first to receive sun. They also have a water film coating so that they can diffuse gases like carbon dioxide into the Palisade mesophyll cells. The spongy mesophyll cells carry out photosynthesis, but they do have fewer chloroplasts in comparison to the Palisade, and they allow gases to diffuse freely throughout the leaf. They are loosely packed with many air spaces in between, and they also have a water film so that gases can diffuse in and out. And lastly, the vascular bundle. We have two types of tissues here, which is the xylem and the phloem. The xylem provides strength. They have lignin deposited to the walls, and we actually learned this in the earlier topics. And their main function is to transport water and ions so that the leaf cells can carry out photosynthesis. Cells are stacked up end to end from roots to leaves. They are narrow, hollow, and they are dead. So no cytoplasm to obstruct flow of water and ions. And these are actually points that I took from our earlier topics. I think that was topic 1, specialized cells. Phloem also has a transport function, which is to transport dissolved sugar in the form of sucrose, and they help to transport amino acids as well. And they have sieve tube and companion cells, and this is one way you can actually identify them in Picto Micrographs as well. And that is basically all of the main structure of the leaf. So I hope that you guys can follow this well. I'm going to give you guys a worksheet, and I hope that you guys can complete it. Please let me know if you need any help. And thank you so much for listening and focusing today. Stay safe and healthy, everyone.